Apis mellifera, commonly known as the European honeybee. What comes to mind when you think about honey? Winnie the Pooh and his honey jar? Maybe your favorite condiment for a glass of tea? Maybe bee stings? How about nuts? Corn? Chicken? Cosmetics? And then lots of money. The global economy collapsing and starvation. That's what should come to mind, because honeybees account for approximately 1/3 of global food production. That's typically envisioned as 1 out of every 3 bites of food worldwide, at least indirectly produced by the honeybee. Pollinators contribute more than $24 billion to the United States economy, of which honeybees account for more than $15 billion. Our agriculture today is intrinsically dependent on the honeybee population. There's a strong mutualistic relationship between honeybee farming and between honeybees and farming. Bees get their pollen from crops, and croppers get their crops pollinated. The honeybee was cast into the spotlight in 2006 when a strange phenomenon swept across farm colonies and throughout the nation. Colony collapse disorder, or CCD for short, was described by the U.S. Department of Agriculture as a syndrome defined as a dead colony with no adult bees or dead bee bodies, but with a live queen and usually honey on immature bees still present. When winter comes, it gets worse. Since 2006, 33% of colonies have declined every winter. But those dead bees aren't just dying. A normal hive has about 50,000 worker bees. Since colony collapse, they're not just dying, they're vanishing. This left the world asking why and how. Alarmed scientists are diving headfirst into the wonderful world of bees and looking for that answer. They're sequencing DNA, conducting studies on agricultural management and bee handling techniques, and researching any and all related parasites or pathogens. Environmental stress, management, parasites, immunities, and nutrition are all being looked at as plausible causes. When honeybees come into contact with pathogens, they have a robust arsenal of innate and adaptive immunities as well as socialized immunity, a feature which is specific to colony-forming insects. The innate immunity includes cuticles, saliva, grooming to remove parasites, and symbionts, any organism with which they live in mutual symbiosis. Their saliva, much like our own, is a main chemical defense and contains the enzyme glucose oxidase, a strong disinfectant that they use to coat the entire hive. On the cellular level, hemocytes recognize foreign bodies and eliminate them, eliciting a secondary immune response within the cell. The midgut of the bee contains the insect-specific enzyme phenyloxidase, which incites a chain of antimicrobial compounds to be released. If pathogens enter the cell, intracellular defenses signal the increased translation and expression of genes encoding for antimicrobial peptides. This is done through upregulation of the gene, which just means an increase of the expression of the gene within the DNA. The formation of antimicrobial peptides follows as such. Here's our cell. Here's our pathogen. When an invading pathogen breaches the physical and chemical barriers and gains entry into the cell, hemocytes find and eliminate it. The pathogen is coated in signal factors that attract the attention of the hemocyte, which is a type of phagocyte. The hemocyte is on the lookout for anything it can label as foreign. When the hemocyte notices the surface proteins, it's able to recognize it as self or as foreign. Foreign bacteria are eliminated by phagocytosis. The hemocyte engulfs the pathogen and induces apoptosis to destroy it. Both the pathogen and the hemocyte are destroyed. When the cell self-destructs, it sends chemical signals out into the cytoplasm, which travel to the nucleus and signal that there are pathogens present within the tissue. This begins the process of upregulation of the expression of genes coding for antimicrobial peptides. So here's our eukaryotic nucleus. It's the nucleus. And inside we have a strand of double helix DNA. The DNA is going to be transcribed into a messenger RNA strand. That messenger RNA can then travel outside of the membrane into the cytoplasm. In the cytoplasm, the messenger RNA is going to develop a complementary strand of RNA, and then with the help of a ribosome, each codon will code for one amino acid subunit attached to each other to create a polypeptide chain that results in the antimicrobial peptide. The antimicrobial peptides are a key form of social immunity within the colony. The infected colony member is able to pass the peptide to other members of the colony, rapidly disseminating immunity to the rest of the hive. But there's a catch. Nutritionally fit bees are able to produce and receive the peptide because it is produced in the fat bodies of the bee. This is indicative of how poor nutrition or a protein-scarce diet can sometimes fed to bees by beekeepers is detrimental to the colony's immunity. A starved bee will not be able to receive the immunity that protein-sufficient bees are able to. Therefore vulnerable to pathogens which enter the hive. Protein-starved bees are unable to receive a key component of the hive's social immunity and therefore susceptible to disease, much like being unable to receive a vaccination to a disease that is already present and prevalent. In addition to a protein-rich and sufficient diet, a diet full of diverse sources of pollen helps to fortify the colony's immune system. So far, protein or calorie-scarce diets lead to protein starvation and the inability of antimicrobial peptides to be disseminated throughout the hive. Social immunity is negated. When a forager bee leaves the hive to collect pollen, it lands and collects from a wide variety of sources. Every time the bee lands on crops or flowers, it encounters new microbes. Whether these microbes are pathogenistic or harmless, the microbes are packed away into special sacs on the bee's hind legs called corbicula and brought back to the hive. Once inside the hive, the bee empties out its pollen stash and packages it along with nectar into the matrix of the hive walls. Here's the corbicula. The pollen and microbes and pathogens all get packaged neatly into matrices and sealed so that it can ferment into bee bread. When the food of the worker bees, bee bread, is consumed, the bees are inoculated with the new bacteria, either enhancing their microbial flora or introducing its immune system to a low dose of a pathogen, acting similar to the bees as a vaccine works for humans. The greater biodiversity of the bee that the bee encounters on its excursions, the greater the biodiversity of bacteria the hive develops defenses against. The crop stores nectar, which is transferred to baby bees via trophallaxis. This is the first enteric bacteria that is introduced to immature bees and constitutes their microbial flora. The greater biodiversity that bees